Welcome to the AI Research Roundup. I'm Alex. Today we're looking at a paper from the Hugging Face trending list, published on August 8, 2025, about 10 days ago. This paper introduces a new framework that gets strong results on medical imaging tasks, even with as few as 5 labeled examples per class. The paper is titled, Sparse Data, Rich Results, Few-Shot Semi-Supervised Learning via Class-Conditioned Image Translation. And as we'll see later, the results are really impressive. It achieves this using a clever semi-supervised learning approach, which combines a small set of labeled images with many unlabeled ones, using a specialized AI system. Also, the authors shared their official implementation on GitHub. If you check it out, you'll find the full source code, along with detailed instructions on how to prepare the med-MNIST datasets and run the training scripts yourself. It looks really well documented for getting started with your own experiments. Alright, so this diagram shows the core of their method, which is a 3-phase training process designed for situations with very little labeled data. The first phase, labeled A, is the initial supervised training. Here, the system's 3 networks, a generator, a discriminator, and a classifier, are all trained together using the small labeled dataset. Then, in the self-supervised pre-training phase shown in Part B, the system uses unlabeled images to generate reliable labels or pseudo-labels for itself. Finally, Phase C uses those generated images to further train the classifier, enhancing the dataset. Alright, so to really put their method to the test, the authors used a wide range of data. This table details the 11 med-MNIST datasets from their experiments. As you can see, these cover many different imaging modalities, like blood cell microscope, chest X-ray, and abdominal CT scans. The classification tasks also range from simple binary choices to complex multi-class problems. And the total number of samples is huge, providing plenty of unlabeled data for their semi-supervised approach to leverage. Moving on to the core results, this table lays out the model performance across different few-shot settings. These settings range from having only 5 labeled examples per class, all the way up to 50. The top 2 rows show this paper's methods, and as you can see, the ensemble version, SPARSE, achieves the best accuracy in every single category. Looking at the most challenging 5-shot setting, it scores over 66% accuracy, which is significantly higher than the other 6 competing models shown below. This table then provides the statistical breakdown for that really challenging 5-shot scenario. The top right portion shows p-values, which measure if a performance difference is statistically significant, meaning it's unlikely to be due to random chance. The values in bold, especially in the SPARSE row, confirm that its superior performance is a real, meaningful improvement. The corresponding R values also measure the effect size, and you can see they are consistently rated as very large, which reinforces the strength of the results. So, a key question is how often the model should learn from this unlabeled data. This next chart explores that by looking at the average accuracy for different unsupervised training frequencies, a parameter they call mu. The key finding is that a mu of 10, shown by the green line, consistently achieves the best performance across all settings. It provides a significant boost, especially in the 5-shot scenario, when compared to the red line, which represents training with only labeled data and no unsupervised phase at all. So wrapping things up, this work presents a really compelling solution for medical imaging tasks, where labeled data is scarce. By cleverly modifying existing unlabeled images, instead of generating new ones from scratch, the SPARSE framework achieves state-of-the-art results, even with as few as 5 labeled samples per class. That's it for this episode of the AI Research Roundup. I'm Alex, thanks for listening.